are listening to Neurosalience, the IVIM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bannattini. Here, I interview brain scientists of all types and discuss their work, as well as their latest developments, issues, and controversies in the field of brain mapping. Today, my guest is Denis LeBihan. He's a clinician and physicist and a relentless innovator in the field of MRI and fMRI since the late 80s. And also, as we hear in this podcast, is a very broad, deep, and highly creative thinker who remains passionate about his work. Denis is currently professor at Neurospin in Orsay, France, and spends time as a guest professor at the University of Kyoto and the National Institutes of Physical Sciences in Okazaki, both in Japan. Denis has achieved international recognition for his truly fundamental contributions to the development of diffusion MRI, diffusion tensor MRI, and the concept of intervoxel incoherent motion to image perfusion. It's actually the B in his name from which the ubiquitous B factor in diffusion comes from. He has more recently demonstrated the ability to image brain activation related diffusion coefficient changes. Denis completed his training in medicine and physics in Paris. He received his doctorate in medicine in 1984 and his doctorate in physics in 1987. He's a full member of the French Academy of Sciences, the Academy of Technologies, the Academy of Pharmacy, and is a corresponding member of the National Academy of Medicine. For myself, his early work suggesting that intervoxel incoherent motion was sensitive to perfusion in the brain played a major role in inspiring my own journey to image brain function with MRI. In this podcast, we revisit this history and we talk a little bit about his work as it evolved over the years and his discoveries and developments as they continued on. And we also talk about his recent work on modeling brain function. He's sort of foraying into modeling, and he's come up with some very interesting and creative models. And he's actually even writing a book on that right now. So I hope you enjoy the conversation as much as I did. Okay, well, thank you, Denis LeBihan, for coming on our podcast. It's quite an honor to have you. And we've known each other since the very beginnings of fMRI. I think we first interacted in the early 90s when fMRI was just beginning. And I think I knew of your work even beforehand. And we'll maybe get into that in the discussion, as you put forth your model on intervoxel incoherent motion and went from there. And you've been sort of pushing the field throughout your career. And so I just want to start out with the beginning. Just looking at your far distant background, did you have any sort of early experiences or mentors that influenced your trajectory in your research and how you got started in that sense?
Wow. Okay. First, thanks, Peter, for inviting me. It's a great honor to share my experience. I feel old now because I have to dig well in the past. But yeah, of course, I had mentor. I would say my first mentors, if I can say that, are my parents. They not intellectually about my research, but they were working in a factory, but they believed that I should learn. So they paid for a subscription in the municipal library. So I would go there once a week. And by curiosity, because I was offered for Christmas when I was about 8 or 9 years old, chemist box. So this is how I was hooked to science. And the box, was not enough for me. So I went to read all the books on chemistry on the shelf. And when I had finished with that, I look at on the next slide. And that was let's say weather forecasting, and I read all the books. Then I went to photography, to electronics. In fact, I built a radio receiver by myself when I was a kid. And so this is my first mentor is the local library, where I learned almost everything by myself. Maybe I'll talk later about weather forecasting, but this is how I got hooked to physics.
Yeah.
Then later on, I was not sure if I would go to neurosurgery, which was my aim, or physics, because I loved physics so much. And it was painful for me to make a decision. So I decided to go to medical school, but after 1 year, it was very boring. So in parallel, I went to a physics degree, so BSMS, to my thesis. And at some point, I was a resident and I was also working on my physics thesis in time sharing. I was very lucky to meet. We had a trip to CERN in Geneva. And then I met great scientists, and I was especially impressed by Sharpak, who later got a Nobel Prize. But he was working on medical applications of physics, and he said, "Given your background, please don't switch to physics. Stay at the top, at the frontier between biology or medicine and physics." And in fact, that's exactly what I have tried to do as the best I could. I hope. But this was the key advice. Then I have been extremely lucky because at the beginning of my residency, I met a neuroradiologist, Professor Cabanis at that time.
Yes.
I remember very well, it was in 1980. And he said, "Well, you are trained as a physicist. Could you tell me what NMR is about?" "Wow, why are you studying NMR?" He said, "Because it seems that we might be able to make images with that." I had no idea about that. And this is how I started to be involved in the French very first MRI system. So he was responsible for checking the medical potential of MRI. And so there was a French company called CGR, which was located not far from Eurospin, in fact, in Buc, which is now a GE headquarters, because GE bought CGR at some point. But so I was introduced to this facility, CGR, and I had access to the machines, and this is where I started diffusion MRI. That was in 1984, 85. And the other mentor, which was very important for me, was Professor De George, because I know in another semester of my residency, I went to neurosurgery because that was, after all, my goal. And I don't want to say he discouraged me, but he said, "Given what you are doing, I think you will waste your time as a neurosurgeon." So I was afraid. He was very kind to say, "Well, you're not a good neurosurgeon and you should do something else." But no, he said, no, you are doing well, but you will be on call every 3 days, and it's a waste. So then I was completely sure. And with MRI coming, at the right time for me, it was a perfect gift to combine biology, medicine, and physics. So these are my mentors.
Yeah, that's actually really, I remember myself sort of struggling in some way against, to either, medical school or like you said, pure physics. Being at that interface is I find, right, exactly where things happen.
In my case, it was really pure physics because my training was in particle physics and theoretical physics. So it was not imaging.
Yes. Yeah, trying to find, physics sort of permeates everything, and trying to find where it permeates seems like where you get a lot of good pay dirt. And
all right,
let's go into diffusion imaging. And this is obviously where you made a massive impact early in your career. And, as I, looking at the history, the concept, I guess, of diffusion was of imaging diffusion was around, I guess since the 60s with Siskel and Tanner. And of course, then there was NMR, but you actually made the first diffusion images, right? As far as that's concerned. So what was that process?
In fact, it's a funny story. As I said, I was working in the CGR factory where I had a 0.35 Tesla MRI system. And one of the radiologists, pediatrist radiologists, said, in the liver, we are not sure sometimes when we see a lesion, if it is an angioma or a tumor. And of course, the treatment is completely different. There was no contrast agent at the time. Yeah. And I thought, I knew the work of diffusion in the past. And to myself, I said, if I have a way to measure diffusion in the liver, in the tumor or in the lesion, I would expect that in angioma, water molecules will diffuse very fast, while in a solid tumor, diffusion will be slower. So I knew the literature. And in fact, most of the literature started back in the 50s to fight diffusion. Diffusion was the enemy because diffusion was destroying the quality of the signal. So
yes.
Hahn and Purcell designed ways to eliminate diffusion.
Yes.
Then Stejskal and Tanner came and reversed the idea, said, well, diffusion might be interesting. So they developed the famous Stejskal and Tanner sequence. And that was mainly Stejskal, I think, with Tanner as a postdoc. And then Tanner took it to look at some excite samples. So he introduced the potential of diffusion measurements with NMR to biology. But with that, you could not do anything in vivo. It's not easily and not localized. So my idea was to localize. So I used this machine from CGR, and I was lucky I had a collaborator called Eric Breton, who was an engineer there. This is how I could get access to the proprietary code of the machine, as all manufacturers, they have their own code. I mean, I could program the sequence. It was in Fortran. It was at that time. It was not difficult, but there are some secret parts.
yeah. I mean, it's always an art.
So this is how I could have access to that. So. But then there were several difficulties. One difficulty was that we could not use the Stejskal-Tanner equation. I know sometimes people talk about Stejskal-Tanner equation, but this is not good because you have to take into account the many gradient pulses that you have for imaging. And they are cross terms, so it doesn't work. So I had to rewrite a Stejskal-Tanner equation, which is a double integral. It became complicated. So because of that, I said, radiologists will not like it. So this is introduced the B value, and the B is from LeBihan, my name.
Yeah. How many people probably realize that? But yeah.
I don't think many people know. But this is, I say, okay, instead of this double integral of time, with time, I call that B. Then you have BD, like for the echo time and NT2, something similar, very simple to understand.
Yep.
And then the second point was that I knew, we'll talk about that later again, but I knew by heart almost Einstein's paper from 1905 on the Brownian motion. So he showed how Brownian motion and diffusion was basically two ways to see the same phenomenon. And in his article, in fact, he shows that diffusion is basically microscopic, and Brownian motion is microscopic. So this work of Einstein is bridging two scales. So that's exactly what I wanted to do, to see with our coarse images. At that time, the resolution was, let's say 2-3 millimeters, what was going on in the tissue in the microscopic level. So it's like a biopsy, virtual biopsy. But then I realized that Stejskal-Tanner equation, the basic equation, is about free diffusion.
Yep.
So I said, I knew Tanner work on restricted diffusion later, but it was awfully complicated. I checked all this work, of course, but I said, again, radiologists will not like it. Let's make it simple. So I said, let's apply the Einstein's equations, assuming free Gaussian diffusion, but then we know that the result will not be the true diffusion coefficient. So this is how I came to the second big concept, the ADC, the apparent diffusion coefficient, which means that what we see is not the true diffusion coefficient, it is apparent. But this is what makes diffusion MRI so useful. If we had free diffusion like in a glass of water, there will be no interest at all. So these are the two main concepts that are introduced at the time, but also it was tricky in terms of corrections for eddy currents, motion artifacts. There were many issues which are still not completely solved today. In fact, of course, we have made a lot of progress. So this is how it worked. So in fact, it was very quick. If I remember well, once I had the idea, it was implemented just in a few weeks only. I was the first volunteer, but today you cannot do that.
Yeah, exactly.
I jumped in the MRI machine and I got images of my brain. I measured the ADC and it worked very well, because in the liver it was a disaster because the liver was jumping up and down with respiration. So, but in the liver it worked very well. Then we had our first patients. They were kids, unfortunately, with tumors and so on, because I was working with this physician, and it worked so well that we published the very first abstract on diffusion MRI in London in 1985, the SMRI meeting. There was no I, it was SMRI at the time.
Yeah.
And we patented it. We had the first patent and several patents. But to be honest, I have to say that in 85, when I presented this work, there were two other abstracts on diffusion MRI. One by a German group, Dieter Merbold. I remember very well because he was sitting next to me where we were putting our slides, and we were looking at each other screen. oh. But he was using a steam sequence, and I think he had images of the hand, but this didn't work, and he stopped. And there was a poster by Taylor where they used something close to my method on eggs, but boiled eggs.
Okay.
And then they didn't do anything with it. So that was the story.
Yeah, I remember Merbold. Was he working with Fromm at the time, or was he?
Yes, with Fromm. Yes.
Yeah, and then you mentioned actually so early work. Also, so Mike Mosley actually did this on
yes.
But it's interesting because the diffusion coefficient it went down with ischemia, I guess, and that's
yes. In fact, this is very very important and crucial for the development of diffusion MRI. Because when I talked about diffusion MRI, people look at me and say, okay, well, and what do you do with it? And it's complicated. So people, they had already a hard time to understand T1 and T2, but now there was diffusion. But what made really diffusion MRI emerge was finding the discoveries by I think we could call them discoveries. I had developed, maybe we can talk about that a little bit later, this IVIM concept that diffusion MRI can also look at perfusion and blood circulation. So Mark Mosley was working on stroke, acute stroke, and he had a model with cats. And he used my method, which I had developed a few years ago. I think it was 3-4 years before that I had developed diffusion MRI. And he tried to see the effect on perfusion, of course, because that's what it is about stroke. And then he noticed something which was weird, that it was really the diffusion, the ADC, which was dropping. That was something new. Nobody knew about that. And again, without the localization. So if you had just the Stejskal and Tanner NMR method, nobody would have seen that. Nobody. So it was because of the localization. You could see the area with the stroke. And it started everything because from that, we had a way to make a diagnosis of acute stroke. As a neurosurgeon and also a neurologist, at that time, even before CT, we were guessing about the diagnosis. Anyway, it was too late to do anything. But at the same time, the RTPA, so the thrombolysis, was already used for the heart. So we had a tool to objectively see the patients at a very early stage, so within a few hours, and check the efficacy of the treatment. So this was done at the MGH, but this is how today patients can get acute diagnosis and hopefully be saved from a disaster, even from death, using thrombolytic agents. I had a talk before this interview with some colleagues in France, and they use diffusion MRI every day. So that was something. And the second discovery of Mike Mosley, I think also by chance, he found that first in the spinal cord, there is a gray and white matter. It looks like an H.
Yep.
It discovered that diffusion was depending on the direction of the measurement, so anterior, posterior, and so on. He discovered that there was anisotropy of water diffusion in the brain. Tanner and Stejskal had observed anisotropy in excised muscles, but in the brain, that was completely new. And, this is how tractography emerged a few years later. So Mike Mosley, I think, maybe not in terms of the technical aspects, but in terms of what made diffusion MRI going to the clinical field were those two discoveries. And the third person that I want to mention, because I moved to NIH in 1987. I was invited, and they got a French CGR machine at NIH in radiology.
Yes.
And basically, I was the key coming with a machine.
Okay. Oh, I didn't know that. Okay.
So I came with a machine. And of course, I started diffusion MRI there, but it was still my spin echo 2D sequence. And 1 year later, Bob Turner came, but he was coming from Peter Mossfield's lab,
Yes.
in Nottingham, where they had set up EPI. So together, we designed this EPI sequence to measure diffusion. Bob built a gradient coil for that. It was really from scratch.
Yeah.
There was no gradient coil, no sequence. And we got help from Joe Meyer from GE at the time.
Yeah.
Because at NIH was a GE site at the time. And we started and we published the fact that diffusion MRI could be made with EPI as that motion artifact free almost, and very fast.
Which was critical.
During my time, each B value would take 10 minutes. One B value, 10 minutes. And the highest was 100. With Bob's coil, it took 1 minute to get 16 B values. And the highest was 1,000. So it turned everything. So if you mix up the work that I did with Bob Turner about making diffusion MRI useful in terms of practically useful, and the discoveries by Mike Mosley, mainly acute stroke, manufacturers, vendors were pushed by clinicians. Well, we want that. And this is how it developed over in the 90s.
Yeah. Well, that's actually a really good point also regarding echoplanar, because, everyone was thinking, it can capture high speed like heart or whatever. But I think people should realize that there's a stability that comes with echoplanar imaging. And even to this day, single shot diffusion imaging is still, everyone's trying to do it with single shot. Multi-shot is very challenging as far as
absolutely. Yes, that's right.
So before we get into maybe starting with the time that you're at the NIH, before we get into the IVIM, which is an extension of diffusion imaging, let's just talk a little bit about your time at the NIH. And you were there, you're working with Bob Turner, and it's interesting. I do remember very well my colleague, actually, Eric Wong, before we developed echoplanar imaging at the Medical College of Wisconsin with his own grading coil, he actually went to your workshop on diffusion imaging. And I think you had a tour of the scanner, and he's like, for the first time in my life, I heard echoplanar, and it sounded so different. It was like this pinging.
Yeah. Yes. So in fact, I have many things to say. I stayed 7 years at NIH. I was supposed to stay there, in fact, and then I would today work with you. But it was a great time, but not only for me. I consider this as a gold time of MRI, because fMRI also came at the same time, DTI, everything. With all this knowledge in 1990, so it was very early because I invented diffusion MRI in 5 years earlier. We had the very first diffusion MRI workshop at NIH. In fact, it was in the Marriott Hotel nearby, but I invited many of the top scientists at the time, including Stejkal. I was so honored. Yeah, Stejkal came. He died some time ago, unfortunately, but such a great guy. He wrote me a letter by hand to say, I accept your invitation.
Oh, wow.
So we had a very nice time. And yes, this is, I remember the great names of this. I still have the flyer of the meeting and the program. Yeah. So I was very proud of that. And because Mike Mosley, of course, was there, and we were all excited about his work on acute stroke and anisotropy. So back to my department, I was in the radiology department, the clinical center. I had the collaboration with a young radiologist from France there, Philippe Douek, and said, well, if we can measure diffusion, let's say, at least in two directions, we might have a way. So the goal is not to measure the anisotropies. From the anisotropy, we can estimate what is the direction of the fibers. Yeah. So if you measure diffusion vertically and horizontally, if the diffusion coefficient is higher horizontally, that means the fibers are more horizontal than vertical. So we published this paper, and the title is color mapping of white matter. In 1991, in JCAT, Di Chiro was the editor-in-chief at that time. Giovanni Di Chiro was at NIH. So it was the very first, in fact, spectrography paper. Yes. But then it was, we had only two directions. And then I met totally by coincidence, Peter Basser. Yes. So NIH, I don't know if they are still doing that today. They had NIH-wide science festival once a year or once every 2 years. Yep. And they invite people to give talks and to give posters. So I was invited to give a talk on diffusion MRI. And Peter had a poster on ionic fluxes in tissues. And we had a discussion. Peter was very excited about diffusion MRI. And he said, what you do is not good because diffusion is not a scalar and it is a tensor. That means that if you change the axis, you will get a different result. I said, of course I know that, but what do you want me to do? Then he started everything. And with Peter, we devised a way to weight, basically to measure diffusion along at least 6 directions. Because the tensor has 9 coefficients, but due to the symmetry, only 6 are necessary. So if you measure diffusion in 6 independent directions, you get a set of 6 equations, and you can estimate the diffusion tensor coefficients.
Yes.
So this is how it came, and we had a great postdoc, James Mattiello, who is also, unfortunately, not with us anymore. But he was great doing the experiments and also making all the calculation, because now the cross terms have to be considered between perpendicular axes. So it becomes really messy, especially with EPI, where you have pulses everywhere. So the B value got promoted as a B matrix. And we published a paper on that. And unfortunately, we were not allowed because of NIH rules to scan human subjects. I was not allowed. It was possible, but not me, because I was a French doctor, and I guess French doctors are not good enough. I don't know why.
NIH rules.
We were not allowed to scan human volunteers, so we scanned some excised tissues. I remember, I bought some rabbit meat from a giant store nearby, and this is how we could develop the very first experiment. Because there was too much rabbit. So I said to my colleague, no, not Peter Basser, another colleague, maybe you could take it home and have it in your fridge. And he said, no way, my wife would never accept to have a dead rabbit in the fridge. Because in France, we eat rabbits, but not in the US. So that was quite fancy. So we developed with Peter Basser DTI and patented it also, and it was extremely successful. And in 1994, I was offered a job in France, and I hesitated a lot, but I decided to accept. I know Peter was very sad, and then he worked with Carlo Pierpaoli, who I don't know if it is because he was Italian, but he has a permission to. But I think that maybe some US license, I don't know what, but he was available for Peter, and they contributed together great work. And I continued from DTI. And from DTI, then emerged tractography.
Exactly, yeah. Yep.
We could talk a lot about that because there are some tricky issues. It's merging between physics and biology and computer science and mathematics. I mean, the very early, very well, of course, the very early algorithms were taken from artificial vision, sometimes completely ignoring what we were measuring. So basically, they would take for granted what we get with MRI, with DTI, and then build tracks using softwares. And, today it works very well, of course, but we know that there are biases, there are artifacts, there are tracks which are not real. When fibers are crossing or whatever, it's tricky. Yep. So there's still a lot of discussion about what we see with tractography.
Yeah.
So it's the end of it.
Yeah, it's an ongoing issue. Actually, I'll be talking with people in the following weeks about some of those sticky issues with interpreting tractography. Although it's incredibly useful.
Oh yeah, definitely. Yeah. It's beautiful. I mean, I know at some point in Paris on the metro station, they had put images of fiber tracks with colors. I was so proud.
Yeah, it's one of the things from MRI that strikes you. If you have really beautiful fine fiber tracks, it just it looks very biologic, but it's also extremely detailed and
yes, and in 3D in colors.
Yes.
And when you think about it, again, most young people would take it for granted. Some people don't even know how it is done with DTI or more advanced techniques. But all of that is Brownian motion and diffusion. It is Einstein's paper from 1905. Yeah. I mean, it is incredible. Einstein would have been amazed to see that his paper gave.
Yeah, and the right, for people just who are listening, who are the basic idea, right, is that yeah, you have diffusion that's restricted in different directions, and depending on the fiber track, and
yes,
longer in one direction than another.
Yeah, that's Einstein's paper.
Yeah. And it's always interesting. And there's different games you can play as well. You can either increase the diffusion gradients or wait a certain amount of time, and that gets different types of,
Yes.
Which is also it's still kind of an interesting topic in some regard there as well.
Yeah.
But let's go on. So that was your major. I mean, that was a huge contribution. You could have, you stopped right there, but you didn't. And
No.
And I first was aware of, early on when I was just starting graduate school, I was aware of your diffusion tensor work, and it was really intriguing. But I was still trying to image brain function, and I remember reading your papers from the late '80s and the early '90s on taking diffusion and applying it to capillary beds, the whole model for intravoxel incoherent motion, and that was also to me incredibly exciting. And a really elegant model, and the way you laid it out in radiology, I think it was the Journal of Radiology and other places, was just beautiful. I don't know if you could talk about that a little bit. Yeah,
but then we go back again to 1985.
Yes. I mean, there's all this stuff in parallel. Yeah,
in parallel. So very quickly, looking at my own brain and then the brain of the patient, I said, I mean, the measurement I had for the ADC were a little bit striking. And I thought that there was some correlation with what I knew about cerebral blood flow and volume in the brain, especially between the cortex and the white matter and so on. So then, again, extremely quickly, looking back at Einstein's paper, I mean, this is my Bible. Okay, I will tell you later there is another Bible from Einstein. But anyway, let's start with this one. And that the diffusion coefficient is, you can estimate the diffusion coefficient of diffusing molecules from the distance between molecules or atoms. So the molecules are bouncing to each other, and the diffusion coefficient is basically the product of the distance between two molecules and the time. So I said, well, if we consider now that blood flows in capillary segments, which are pseudo-randomly oriented in space. So then we have a kind of random walk. That means the flowing blood goes from one segment to the next, and there is no correlation between the directions. It's sequential directions. So this is a random walk. And I just copy-pasted Einstein's equation. I'll say, but let's now call the distance between two molecules the segment, the capillary segment length. And then I had the velocity of the blood. Of course, the order of magnitudes there are 5 orders of magnitudes in difference. But when you calculate, when you mix together the distance and the time, and you get another diffusion coefficient, which I call D star, so the pseudo diffusion coefficient, it's only 10 times larger than the true diffusion coefficient of water. Okay. So when I found that, I said, that's it. Now, I know why I see it with diffusion MRI, because it's so close. But I knew, and that was my second paper in radiology with Tom Dixon as a reviewer. He gave me his review recently. It's amazing. And I said, then we have a way to separate D star from D. So we could make images of perfusion and from the IVIM effect, different from the diffusion images. And my goal in 1985 was the same as you, was to do fMRI. So because with PET, they look also at the blood flow, right? Say, well, I have with MRI a way, without injecting any tracer, a way to see if there is an increase in blood flow. So I did the experiments on the daughter of my physics PhD thesis mentor. So she came to the facility. And I remember I was doing a visual stimulation, flashing in front of her eyes, some image, and so it was very crude. I think I could see some effects, but I was not very convinced. But I still have the images, and now looking at them, I think there was something, but it was very. I could not make anything with it. And then I came to NIH. Yeah. And I was not working on that anymore. But Alan Song at Duke did it, and he showed that it's working. You can do fMRI with the IVIM effect.
So let's dig into that just a little bit. Yeah. So so let me just back up just for a second here.
Yes.
So with the IVIM, I mean, certainly if you just had capillaries, right? You would attenuate that. But, I remember at the time, there was still always worry that, it attenuated maybe CSF, or it was very sensitive to motion, or even intravascular large vessel signals, you might dephase that more rapidly. And so I think that there was some issue with there. I mean, certainly it's been worked through, but I don't know if you want to talk about some of those things that came up. Like, people would be worried, like, what are we looking at here? Is it just CSF pulsation, or that sort of thing?
Yes.
How do you separate?
Yes. Yeah, in fact, in my 1986 article in Radiology, there is one case where you see a CSF flow beautifully with it's an IVIM effect, but it is CSF.
Yes.
This is how I could say we are not looking only at diffusion, and this is why we call this, with my mentor in physics, intraboxal incoherent motion, to say that beside diffusion, anything incoherent, so not head motion is coherent. People had a hard time at the beginning to understand that. Yes. But yes, it was tricky because there was this caveat. Also, we see it only at very low B values, and the effect is very small.
Yes.
So at the 1990 workshop I organized at NIH, it was on diffusion and microcirculation with IVIM. And there were some of my NIH colleagues from the same place, like Rick Boonen and James Picard, who were wearing T-shirts. On them, they had written diffusion, perfusion, confusion. I was
oh my gosh. Oh, wow.
In front of everybody at the workshop.
Oh, great.
You have to be brave to not to give up,
Yeah. Tough crowd at the NIH sometimes, right?
Well, it was fun, but sometimes you ducked. And and at that time I was not doing any animal experiments. So it was difficult to prove that. I was sure I was right because of other pieces of evidence I had, but it was hard. In 1999, there was a very first paper, a clinical paper in the liver by a Japanese team showing that indeed they could answer my question about angiomas and tumors in the liver from the IVIM effect. So it was there. And after that, it increased a lot. And today, it is used extensively for cancer in oncology, because you have a way to measure blood volume and blood flow, mainly blood volume. And in tumors, in malignant tumors, there is angiogenesis, and the proliferation of small vessels. So you can watch that in time, and you can also check if the treatment is working, because the treatments are also working against the small vessels. So, and I think 3 years ago, we published a 500 pages textbook on IVIM MRI with my colleague, Dr. Ima from Japan, Dr. Fedro from Switzerland, and Dr. Eric Sigmund from NYU. So you see, it took a long time. So that's the problem. I mean, diffusion and IVIM and everything I'm doing, it looks like it takes 10 to 20 years before people realize that it could be useful. I'm getting old. So some instance, you 2 years later, great. But in my case, people at the beginning, I'm not sure. I don't understand what you want to say or to do. And before people realize that not only it makes a lot of sense, but it is working and useful, 10 to 20 years. I think diffusion MRI, DTI 10 years, IVIM 20 years.
Yeah, and it's such a rich area. I mean, have you even considered it? It's just occurred to me. Have you ever considered trying to make apparent or IVIM generated tensor maps?
Yes, in fact, I had a PhD student, Gabriel Foray, a few years ago, and we, because in my radiology paper from '88, I introduced two extreme models One model is the exponential models, which basically exactly the same as diffusion and Einstein's equations. But if the flow in the capillaries is too slow, or if capillary segments are long, then the blood will not change direction.
Right.
Then if you do the calculation, you find that the distribution is a sink curve. But in between, it's more complicated.
Yeah.
So there are some PhD theses about that. My student worked out with a mathematician. We had Jing Li with us. And we showed that if you take the sink condition, the condition are for sink, because of the distribution of velocities and the distribution of capillary segments, you end up with an exponential anyway. So the exponential model looks correct, even if we are not in the physical regime corresponding to the exponential. But we look also at the anisotropy, because especially in white matter, the vessels are not completely random. They follow the tracks. And we could see some anisotropy effect, but most importantly, I think for people who do IVIM, because people doing IVIM and diffusion, they don't care about DTI most of the time.
Yes.
But then in the brain, depending on the direction of the measurements, the IVIM effect will not be the same. Not because IVIM is anisotropic, but because diffusion is anisotropic.
Okay.
So if you fit the data, then you get the bias in the IVIM. So we showed that. And also by changing the diffusion time, you can switch between different models. No, there is a lot to do. Yes, clearly.
Yeah. There's so much. Yeah. And actually, let's just, I'm really curious about, so what I actually tell people, I've been telling people for years, and now with 7T, it's less of an issue. I say, well, if you want to get rid of your intravascular signal, I mean, this is more large vessel intravascular signal, then just apply a little bit of diffusion weighting, and that dephases it. And that's sort of the effect, now when I was doing my postdoc at MGH, and also Alan Song at the time around 95, showed. I mean, so let's go from, like, he showed that, with diffusion imaging, it attenuated the bold signal much more at 1.5 Tesla or 3 Tesla. And then at 7 Tesla, it didn't, just because the T2 star of blood is so much lower that the signal's already gone. But how do you go from, just for everyone to understand, how do you go from what is attenuated in the bold signal? The bold signal is attenuated by diffusion imaging because you get rid of the intravascular signal. How did he then image IVIM from that observation? I mean, was it from that observation that was inferred, or was he actually imaging IVIM?
We measured really IVIM. So it's basically what Alan Song did, because it's not the IVIM weighted. I mean, if you calculate by fitting the curve, the Bold effect, I'll talk about that in diffusion fMRI maybe, but the Bold effect is present in plain diffusion MRI because it's a T2 weighted sequence. So you have the Bold effect anyway. If you manipulate different signals from different B values and you take the IVIM part, you can cancel the Bold contribution. So we know that it is a genuine perfusion effect observed with IVIM with low B values. Now, it is also true that you can clean up the Bold response if you have a gradient echo sequence by killing the signal from large vessels. But that's not the IVIM effect.
Yes. Yeah, that's an important point.
Yeah, so then you can dephase a fast-flowing blood in small arteries or veins because those vessels are more remote to the activation sites. You artificially increase the spatial resolution of Bold fMRI. So I know some people have suggested that. I'm not sure it's used every day, but it is working. That's for sure. But then there is a other effect. What if you now calculate the ADC? So you from B values above 200, and then the IVIM effect is gone.
Yes.
And then you start to see something else, which is a pure diffusion response. So
let's get into that.
Yes, the signal acquired at 200 and the signal acquired with B of 1800, the Bold effect is the same. There is a Bold effect present. I will never say that there is no Bold effect, but it is very small, especially with the spin echo sequence. But if you calculate the ADC, you find that the ADC is going down during the activation. And that's what we published. I published in 2001 with one of my PhD students at Dake, but more convincingly, I think, when I was in Kyoto. So this PNS paper of 2006, where we showed that the ADC was going down. It is really the ADC because we can show that the value, the effect depends on the B value. So it is really scaled by the degree of diffusion weighting. But what was so nice was to see that the timing of the response was different than Bold and much faster. by 2 or 3 seconds. It's huge, you know.
So it's faster. So I remember when I first saw it, right? It's faster, but it's not like, I mean, I guess it depends on the sampling rate of MRI.
Absolutely. Yes. So that's right. In fact, at that time, I think the TR was 1 or 1.5 seconds. Now we have switched to line scan fMRI. And so we sacrifice one spatial dimension. So we have only one line, but then we can go to, let's say, 250 milliseconds. And then we see the time course. And we're not the only one. I mean, Daniel Nunes at the Champolino in Lisbon and Noam Shemesh have published, I think in your image, a beautiful paper showing really that the time course was very fast, but also what they've seen, and we see it too. We have 2 peaks. One peak which is occurring during the activation window, and one peak which is after the end of the stimulation. It's not a shoulder; it's a different peak, and we see now with our line scan that it is not at the same place exactly. Okay. We don't know what it is. We had an abstract at the ISMRM this year. My hypothesis is that I believe diffusion fMRI is about neuronal swelling, but there is also astrocyte swelling. And we know from the literature that astrocyte swelling occurs remotely in time and space. So I think that the astrocyte swelling is related to BOLD. So really? We have to pay with diffusion fMRI to dissect the mechanism. So what is occurring early, which is direct neuronal firing and hopefully neuronal swelling, and something which is a little bit later, which is energy restoration from the astrocytes, and that is delayed in time, and it's not exactly at the same place. So it's very interesting. But again, diffusion fMRI, I mean, when I published this paper, many people, it's not possible, it has to be bold, blah, 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 blah. I mean, again, I'm used to this.
No, I was quite excited by it. We actually, I mean, and it's hard though I mean, we tried to see it. We weren't that successful, and maybe just because there's so many details of the sequence. Obviously you're giving up some. It's a little bit noisier. The signal effect is the same, but it's a little bit noisier, and there might be eddy currents or whatever for us at least. And so we had a hard time seeing an effect. Before we get into that detail, I mean, just to clarify for the audience, the basic idea is that the diffusion coefficient within cells is less than outside of cells. And so with activation, you have some cell swelling, and then therefore it's a larger volume of that which has lower diffusion coefficient, and the diffusion coefficient goes down. And it's only measurable at very high B values because it's overall very slow. And that's the basic model, and that's still agreed upon. I remember
that's maybe not exactly what the model I have in mind.
Okay.
Now, we've done additional experiments. Well, first, because I was, quite attacked about diffusion fMRI, which has to be bold. I mean, it is a
well, there is that one paper by Carla Miller that was pretty. I mean, I looked at that and I thought, oh well, that sort of maybe suggests it's vascular, but then there's obviously ways of.
I know this paper. If you look at the curves she shows, she tried to increase CO2 in volunteers, but if you look at the curve, it's only the very last point where you see something maybe.
Yeah.
I have a lot of respect for Carla. I know her very well. But honestly, I don't believe in the data she published. Okay. I think it's anyway. But that's good because she pushed us to the limits. So we published some papers in animal models showing that if you kill the neurovascular coupling with nitroprussate, which Ogawa did for BOLD, the BOLD response is gone. But not the diffusion response. It is remaining intact, which means that it has nothing to do with the BOLD response and the hemodynamic response.
Well, let's just wait a second. So if you kill the neurovascular coupling and you have an increase in metabolic rate, wouldn't the BOLD signal go down, and it look like? I mean,
the BOLD signal disappears, and you might see in some points a negative BOLD response because then you see the oxygen consumption. So we see that very well. So the positive BOLD response is abolished, but sometimes you see a negative response.
Yeah.
But for diffusion fMRI, nothing.
Okay.
No change at all. When you look at the ADC, if you look at the diffusion weighted image, then of course the BOLD effect is gone. So you have a small effect, but the ADC, no. So, I mean, hopefully this is highly convincing that it has nothing to do with the hemodynamic response.
Right.
It has to do, Second a piece of evidence we've published also in PLOS Biology. We are awakening rats under anesthesia. We'll talk about that with a new model I have of the mind. By stimulating a very small nucleus in the thalamus, you can wake up the animal. So you have to send some electrical current at a given threshold. If you block swelling there, if you inject locally furosemide, then you have a really hard time to wake up the animal. You have to increase the threshold a lot. You induce the swelling by using a hypotonic solution, so diluted CSF. Then you can wake up the animal with a very small current.
Wow. Okay.
And at the same time, we measure the ADC, and you see the ADC locally. The decrease in the ADC is there, but remotely also. So you see all the areas, including the sensory motor cortex, which are the areas which are linked to this nucleus in the thalamus. They see the ADC changing also when the rat gets awake. And we are not injecting anything in those areas, remote areas. So you see that it's really amazing that cell swelling, I think, is really the mechanism of diffusion fMRI. Yes. Now, to go back to your very interesting question, why a cell size increase, so cell swelling, would lead a decrease in ADC? But this goes back to acute stroke. In acute stroke, we have a big drop in ADC because of cytotoxic edema, cell swelling.
Yes.
So to answer your question, we did also some work, which published. We look at single neurons, a diffusion fMRI of single neurons. So we cheated a little bit because you look at huge neurons with the Aplasia californica model at 17 Tesla. So then we did so we can see the neurons one by one, and we can also look at the signal in the ganglion. That means by taking all the neurons together, right? We can look at both. So what we see when we stimulate the neurons with dopamine, first with our eyes on the images, we see the swelling. So it is there, period. Yes. When you put the dopamine there, you see the neurons swell. Okay? Yeah. There is no blood, nothing, just neurons. So cell swelling is not a fantasy. It's right. It's there. Now, the ADC inside the cells goes up, not down. That's exactly what we expect because you have a dilution.
Right. It's bigger, and so there's more.
Yeah. So the ADC goes up, not down. So then why is the ADC going down with diffusion fMRI? Now, if you take the ADC on a region of interest, including several neurons, let's say a dozen of neurons, and the extracellular space between them, then the ADC goes down, and the effect is larger than what you see for the increased ADC inside the cells. Now, we did some calculation because we can also measure the extracellular space. We can see it. So immediately you say, well, the ADC goes down because there is a shrinkage of the extracellular space.
Yes.
That's not enough. I mean, it doesn't work. So what is left is my favorite model, which I published also, which is that we have a layer of water molecules bound to the cell membranes because of electrostatic charges. So the big debate is how far does that go? Is that only 2 layers of water molecules or 100 layers? There is a big controversy in the literature. I published a book. It's called Water, the Forgotten Biological Molecule. I invited authors from both camps. And some told that I don't want to write a chapter in your book if this guy is writing a chapter. That was, I mean,
politics. Yeah.
But I used my charm or whatever, it worked out. So they all contributed. So this is the idea. So there is this layer. So if you increase the surface of the membrane because of stroke, because of activation, you increase the fraction of this slow diffusing water. Okay. So that's why the ADC goes down. But of course, water molecules say they don't care about your model. They use anywhere they want. So there is exchange, and exchange is usually completely ignored in diffusion MRI physics. This is a big error, I think. I mean, water molecules, they are not free, but they can cross the membranes. And we show that, in fact, they cross a lot. We've showed that with a different kind of setup. So if you have water molecules diffusing, so they spend some time in the intracellular space, sometimes in the membrane-bound layer, sometimes in intracellular spaces. It's a balance. So what you see with diffusion MRI and diffusion fMRI is the proportion of the time they spend in this layer. Okay. So if you take the surface, all the surface you have in the neurons in the visual cortex, in the human visual cortex, it's huge. It's huge. I mean, and even if you have let's say a 2 or 3% increase in the cell volume, and only 10% of the cells which swell, so 10% are activated, then I model that it is explaining what we see in the human brain.
And
I don't want to say
more of a surface area. So there's more of a surface area for which spend time around that.
Exactly, because there are so many spines, and each neuron have, let's say, 10,000 dendritic spine, and they're all swelling. I have movies in cat models where you see all these spines swelling and shrinking all the time. It's completely dynamic. It is there. It is there, but people, I mean, they love bold. I like bold, but right. If we can get a more direct signal, and I'm sort of agnostic. I mean, I just want to know what can be done and what's interesting. And I tend to believe it, and I think that if we can somehow clean up the noise, there's all these techniques that are coming out. You're right.
Might be able to help.
Yeah, the point. The amplitude of the diffusion ephemeral response is the same as bold, even a little bit bigger. But the spin echo with long diffusion time, because we cannot put the gradients otherwise, is killing us. I mean, the SNR is not as good as bold. Yeah. So my conclusion is that if you want to do cognitive neuroscience, bold is good, is perfect. It is easy and no problem. If you want to do, I don't want to have bad words, but neuroscience, you know what I mean. Understanding the neuroscience of activation and so on. So you are interested in the spatial resolution and the temporal resolution, then diffusion fMRI would be better, I think. And especially if you work with animal models.
Yep.
Anesthesia is a killer with bold. I mean, we've looked at medetomidine and isoflurane. They have opposite effect on the bold response. Yeah. While for diffusion, no effect at all. If you want to do resting state fMRI in animals, I mean, you have to consider the effect of the drugs.
Yeah.
Well, with diffusion fMRI, you don't care. So
and that's actually another thing that occurred to me too, is that you could potentially, I've been trying to look at, using MRI to look at, global changes in inflammation. And it's sort of like starting to bubble up in the literature. It's sort of saying, well, maybe global inflammation with lifestyle or whatever, lack of sleep leads to Alzheimer's or whatever. And so trying to use MRI to look at global inflammation, it seems that there's a way with diffusion, with this sort of trying to get a global assessment of this in some sense. But
yes, I agree.
But also there's a big thing that everyone's really intrigued on, and that's looking at the lymphatic system.
Oh, yeah.
You've been looking at that. You've been starting to look at that with a number of papers too.
Yes.
So might want to. Yeah.
It is intriguing. I have to say that it is a fascinating field because it might explain many things up to Alzheimer's disease. There are models for that. It might explain why gadolinium get deposited in the brain. It many things. But so I'm not an expert on that, so I don't want to talk too much. Yeah. But the idea is that the astrocytes are also playing a big role there. And we did some experiments where we manipulated the astrocytes, blocking the swelling of astrocytes, for instance, and so on, with TGN20. And we've seen that with diffusion MRI, with something new I have introduced recently, which is the S-index. S-index is a way, it's not the ADC, it's a quantitative marker which doesn't depend on any model. It's just that you look at the signal, it's a little bit like artificial intelligence where you want to classify tissues without making any hypothesis. Yeah. And what we see is that with diffusion MRI and this S-index, we can catch the fact that we can monitor the astrocyte activity, but that's it. So we don't claim anything really yet about the lymphatic system because there are many controversial issues. I'm a strong believer that yes, there is such a system, but then the IVIM effect, considering CSF, my estimation is that no, there should be no effect on the CSF flow in the tissue itself. In the ventricles or in the vascular spaces like the virtual Roman spaces, probably. Some people have claimed that there is CSF flowing in the parenchyma. I don't think so. At least we cannot see it with IVIM and diffusion. We look at that and we don't see anything. So it is open field. Yes, I'm very much interested, and I believe diffusion MRI is a key player there as well, but I cannot say more at this time.
I've often suggested to sort of like try either looking at, looking doing some sort of diffusion weighting to try to pull out the lymphatic system. But it seems like the most successful results have been right with Laura Lewis looking at the fourth ventricle, just slow
oscillation. Yes.
Which is interesting. So, okay, so let's, so this, I mean, right. I mean, and actually I'm definitely excited about using MRI to look at diffusion changes with activation. And I think that I believe your results is just a matter of, once again, pushing the field forward to sort of have less noisy techniques and really be able to get robust findings so people could use it. I mean, I've been trying to look at neural current effects, which are sort of, hypothetical, but not exactly there either. I mean, no, much further back from diffusion, definitely. I mean, but the results. Yeah.
But what you've done yourself about electrical currents is also amazing. And again, it's the same also that the effect, I'm sure you're right. I'm sure this effect is there. Just too small to be catched, to be seen. Exactly. But it might be that when we go to very high field, then we might see something, because you must be right. I mean, the physics with Lorentz forces, it has to be right.
Yeah, and with MEG, you're measuring something, and so you must have a source. To the degree you have a source, you can be coherent. Yeah, and I'm always holding out hope for that. It's still working along. But certainly diffusion is further along the line where you can actually see a result, and now it's just a matter of refining the technique and applying it. And there's so much more degrees of freedom with diffusion imaging that you can actually do a lot with that. So that's great. What about, okay, so now let's switch gears completely. So talking about high field briefly, about 10 years ago or so, or actually in 2007, you started Neurospin, and which I've had the honor of visiting once in about 2014, and I was just blown away. It's a really impressive center. And the idea was to, house a human 11.7 Tesla scanner. And you were the first to sort of like push that. And you actually had to work with the French government to the Atomic Energy and Alternative Energy Commission. And and also you're working with Siemens too, but it was very challenging. Could you describe that process? And
yeah. My time constant is 20 years, as I said. So in fact, this project started 20 years ago, and Neurospin came a little bit later. So my idea, I can tell you the story. It was in 2000, I believe. I was showing to a VIP coming to our center, which was not Neurospin, a small hospital nearby, our 3 Tesla MRI system, because in France, it was the first one. And I was supposed to say I was very happy and blah, blah, blah. And instead I said, well, it's great, but, given my background in physics, and I know these guys at this Atomic Energy Commission in my institution, they make magnets for the CERN in Geneva. We could make an 11.7 Tesla MRI magnet for human studies. So people look at me considering that I was crazy, drunk, or something happened to me. But just over a few weeks, I was called and said, this is such a great idea. Let's do it. And then it's a long story because, again, it took 20 years. But I wrote not even a single grant. I wrote a report explaining my ideas and what would be the point to make such a magnet. And then we had a tender, of course. Manufacturers, they all said, no, anyway, we cannot make something like that. And only CA made a feasibility study and showing that they could, in fact, make copy-paste the design used at the LHC in the CERN for particle physics, with a special double pancake design, and there are many tricks for this magnet, and hopefully make a magnet with us, which was within my specifications about time stability and spatial homogeneity. So in 2004, we had a meeting at the top management of the CA, and we got the green light. Do it. It's a risky project. That was the conclusion, of course. But it is, you do it. I mean, you, but not me only, of course, everybody. So then they worked out the details. It's a crazy project. And in fact, it's not a single Y. It's a collection of 10 filaments to make the Y of niobium titanate. So 11.7 Tesla is at the limit. So in order not to go too far above the limit, the magnet is cooled down to 1.8 Kelvin, not 4.2.
Oh, wow.
Yes, for the helium to be superfluid. That means that if there is any temperature increase that's locally, it is instantaneously dissipated. At the speed of light, but anyways, inside the magnet to prevent a quench. And because of that, the magnet we had to build a cryogenic plant underneath. So at Neurospin, there is a cryogenic plant to produce liquid helium at 1.8 Kelvin continuously to feed the magnet.
Oh my God.
It's something special. So you see, Neurospin came as we need a place to have this magnet. So we decided to build a new research center housing not only the 11.7 Tesla, but other magnets, including one which is only in my brain now that nobody knows about. There is one arch which is empty to welcome a new kind of magnet. So I'm working on that. And the other point is that the magnet is, because it would be too expensive and too complicated, it's not a solenoid. It's a double pancake design. And because of this design, the magnet has to be an external power supply. That means a lot of risk for instabilities. So there are several patents about this magnet to compensate for instabilities and stabilize power supply and everything. So you see, there are more than 1,000 sensors in the magnet.
Yes.
And everything is automatically controlled because, in France, we have a lot of vacation, we have strikes, there is a pandemic, many things happening. And to be sure that the magnet is always secured. So, my colleagues had to design also a special system to control the magnet. So the magnet is huge, 5 meters by 5 meters, and 150 tons. And to go from Alstom, where it was built in east of France, which is now GE Alstom, but not nothing to do with MRI and imaging. And Neurospin, it had to go by boat on the Rhine River and the North Sea, and then the Seine River. It took 3 weeks for the magnet to travel. It arrived in Neurospin in June or May 2017. And then it was a very tricky operation to put it back inside Neurospin. Then the cooling system had to be installed and checked, and homogeneity, to be sure we could get to 11.7 Tesla without a quench. We went slowly. I think we went to 11.7 Tesla 2 years ago, and now everything is ready. So Siemens equipment came at the last minute. Siemens. It's a standard equipment,
right?
And so it was installed very recently, and Siemens is now finishing some tests, and we expect to get our first images very soon. And before we get to start human subjects, we have to go to the administration of France to get the permission. And unfortunately, this will take time. And I don't think we can human subjects before 2023. But you have your 11.7 Tesla also there. So you are...
Yeah. Right. At the NIH, we have 11.7, and we're still in the process. Helium is an issue. So to actually obtain it, and it's expensive, and it's coming, I think, it should be here soon. So we should be ramping it up pretty.
Oh, okay. Tell us. If you want some helium, we can ship to you.
we might be asking you. But yeah, no, that was our bottleneck right now, and certainly we were hoping to get it ramped up hopefully by the winter time.
Oh, good. So that's a terrific project, and I'm so proud of that. And about 200 people worked on it. What I didn't say is that, of course, we need money to to build such a monster magnet. So in 2004, I managed to put together a consortium, a French and German consortium of academic partners such as CA and industrial partners like Gerbe in France, SME making contrast agents, and Siemens. And we got approved by we got the money, huge amount of money. I think it was 215 million euros, but not for the magnet, for to develop also new kinds of contrast agents aiming at ultra high field MRI. The magnet is a quarter of that, and this is how I came with the name ESOLT for the project. So ESOLT was because I'm a musician. ESOLT is French and German opera with Wagner and Debussy also in France. So I said this is good, and people accepted it, so it's called Isolde magnet.
Okay. All right. Well, that's it. So one thing actually, and so our scanner actually quenched partially because we were trying to push the gradients, and there was an interaction of the high performance gradients with pushing the duty cycle, sort of heating up maybe the bore a little bit and sort of causing a quench. And then obviously it's being fixed right now, but still, it's been fixed. But your scanner, so even though you care about diffusion, your scanner doesn't have the high performance gradients, but hopefully it will get those, but there's a real risk. I mean, maybe it shouldn't have those just because of the
that could be, but in fact there is a big difference with your magnet. Your magnet is 65 centimeter bore, right?
It's tiny. It's a small.
Yes. While my magnet is 90 centimeter bore.
Oh, then
when I gave the specification back in 2001, people said, "Wow, it's huge. Are you sure it will be so expensive?" And I said, "Yes, it is to minimize interactions of the gradient and the magnet." So when you had your quench, they looked at me and said, "You were right."
I think you might be right. Yeah, definitely.
So thank you, Peter. and the second point is that the lesson what we have inserted between the gradient coil and the magnet a shield. The shield is also limiting the amount of heating and the boil off and eddy currents in the magnet. So this is tricky because but again. It seems to work extremely well, and we are looking over the shoulders of Siemens so that they don't push their gradients too far. But yes, for diffusion MRI, because the T2 and T2 stars will be so much shorter, we might have a hard time to do diffusion MRI. So the idea is to start with the gradient systems that we've got from Siemens, but then develop a more advanced gradient system. So not the connectome, as in Siemens, and especially Franz Schmidt was so, no. I think they said if we quench your magnet, it will be so expensive.
Yeah, that's risky.
Maybe there was some politics, not only science or technology, but I have to say that it is not 3 Tesla, and the forces are huge. So I couldn't understand very well. And if we go to more to faster and stronger gradients, we have to be very, very careful.
Yeah.
And we need to get the money anyway.
or even very small localized coils as opposed to the
you have to be careful. Yeah.
Yeah, no, it's exciting. It's exciting, and these things take time. And
yeah.
Let me just skip ahead a little bit towards the. I'm respectful of your time. Obviously, this is a little bit longer, but it's perfect. It's great. There's a great discussion. Why don't we mention a little bit? Obviously, one thing that has always struck me about you is that you're very broad in your thinking. You're intellectually, fearless in terms of going into various areas. And I noticed that there's a paper that struck my attention in the journal Brain Multiphysics on time and space in the brain, a relativistic pseudo-diffusion framework. And maybe without going, obviously you could spend probably hours talking about this paper, but it was really struck me as sort of like an extremely novel reconceptualization of trying to do brain modeling. And I'm kind of curious about, what motivated you to think about this and where you're taking this idea.
Yeah. That's my last child, basically. So far. Yeah, so in fact, I've got interested in the relativity concept, or relativity theory, back in when I was a teenager, in fact. So it's long. I realized that maybe we don't consider the time properly in the brain. If you want to catch a fly, you take your hand, but by the time you go to where you think the fly is gone. The fly is smarter than you. Why? Because the brain makes an image of the past, and then with some processing decides maybe you are not even aware. Well, don't forget you have to catch a fly, and then you have to move your arm and everything. So by the time you get there, so the brain is making a prediction, a forecast about where the fly will be, but the fly is going to random walk, Brownian motion, and you usually don't succeed. It's the same with tennis. When you play the tennis, the balls are so fast, the brain has no time to calculate. So the brain, based on the behavior of the guy across you, and on his eyes, the positioning of the body, your brain make a calculation about where you have to put, not where the ball will be, but where you have to put your arm with to catch a ball.
Yes.
So my conclusion was that present is meaningless. And when you do fMRI, of course, the time resolution is an issue, but you see many spots activated. But in fact, if you look at the sky at night, you see many stars, right? So you may consider they're all like activated things. But in fact, because of their distance, you don't see the stars at the same time. So all the stars you see have been sending light at different times. So it's the same. So when you look at the brain from outside with fMRI, or even if you have MEG because you have a little bit of the time, you have a view which I think is not what, if you are an action potential flowing in the brain, that's not what you will see.
Yeah. Yeah.
So this is going, and the reason is that there is a speed limit in the brain connectome. So the action potentials don't flow instantaneously between two areas. So if you take two nodes, A and B, and you see them firing exactly the same time, A is not aware that B is firing, and B is not aware. I think these kind of concepts are not very well considered in the current literature. So if A is activated, it's because in the past of A, there was a cascade of activation, right? So in fact, this is exactly the relativity theory of Einstein. And so that's why I said time is a special dimension. So we have to consider not time and space separately, as it is done with current models, but we should consider that the brain connectome has a space-time in 4 dimensions. So at a given point in the 4D space-time, which I call an event, this event is connected in the past, in the future with a cone. That's a Minkowski kind of diagram. And the opening of the cones is set by the velocity of the propagation of the action potential. So it is becoming more difficult than for physics because the speed velocity depends on the length of the connection. As you know, long fibers are more myelinated and they conduct more faster than small fibers. So you have to consider all of that. So it makes things more complicated than the relativity theory. But you can import the math of the Einstein's equation to model that. And in my paper, I said, well, even Einstein said that the speed velocity is a limit, but it could be any limit. So in the brain, it's not C, otherwise it would be very fast to think. I call it C star, okay? It's much slower, but because there is a speed limit, you have this problem. So then now you consider that when action potential flow in the brain, it's like geodesics. They follow the lines, so the connections, which will minimize the energy in the 4-dimensional space.
Okay.
So in order to develop further this concept, I went back to my favorite field, which is Rodenburg and diffusion and IVIM. Yeah. And because you need to write some physical model for that. So again, a D star and everything, but it is not now blood in capillaries. I consider action potentials along fibers.
As the diffusion of information.
Yes, but now you cannot consider the fiber length or the velocity because length and velocity are related to space and time. So it's becoming messy. So instead, there are ways using the kinetic theory. But what is really, really impressive is that when I did that, I look back at the paper from Einstein from 1905, which I thought I knew by heart. And then I discovered that he made, I don't want to say a big mistake, but this paper is not compatible with a relativity paper he published 2 months later. Because there is no speed limit in the diffusion paper of Einstein.
Oh, interesting. Okay. Oh, that's interesting.
So when I discovered that, I was shocked. So it was at night, and the next morning I went to the web, of course, and I discovered that a few guys have catched up this problem. The problem is that the solution is extremely difficult, and it has been only solved recently. There are a few PhD theses on that, because it is used now to model the displacement of galaxies or high particle energy physics. So it's my former field. So if you mix up everything like that, something happened in my own mind. I was lucky a few years ago. I went to a library fair in Kyoto University and I bought 3 books. One book was the geometry of Japanese temples. And, Japan was isolated from the world. at the time. So the Japanese architect had to devise new kind of geometry, but it is extremely interesting. Then I read a novel by Ishiguro, who got a Nobel Prize for literature. It's called The Unconsort. It's a story about a pianist who is supposed to give a concert somewhere in the middle of Europe, but it is really disconcerting. The pianist is completely lost in time and space. He's mixing up different events of his life. It's like he's dreaming, but he's not dreaming.
Yeah.
And this is how I got this space-time ID, basically.
Okay.
And then I had a textbook on relativity theory. I learned it when I was younger, but it was a refresher. I went further. So using this model, so it's complicated. So I use simulation instead of solving the equation. That's good to have computers. And you can show, for instance, that you might explain why schizophrenic people hear voices. We all hear voices. But if the time is wrong because the fiber connections are wrong, that's something we see with DTI. The order of the event in the 4D space-time could be reversed.
Yes.
So they hear the voices before they realize that they are the authors of the voices.
Got it. Oh, that's interesting. So, right, the delays are wrong in such a way that you can actually...
I'm writing a book. I'm almost finished about all these ideas, but it's, the Schrödinger cat who is alive and dead. But if you read in a book that, in fact, the cat should be dead, the schizophrenic patient doesn't realize that he has read it. So somebody asked me to kill the cat.
Interesting.
because he has to invent a story. I mean, he's hearing that the cat is dead, but he doesn't know it was something he read. Of course, it is.
I know.
Explanation. It's more sophisticated than that.
Yeah, so, I totally, and certainly, I recommend anyone reading the paper, and also, I mean, I think that there's like anything. I mean, it seems like there's elements of that glimpses into sort of insights of, I mean, it's a very fascinating model of
and it goes further because that was the special relativity, but there is a general relativity. The space-time is curved by masses and gravity, but it is exactly the same for the brain. So I show that an activated node of the brain has a mass, a virtual mass. In fact, there is a mass if you consider cell swelling, but let's forget that. So you can model. I mean, my pseudo-diffusion, relativistic pseudo-diffusion model is basically assuming that the amount of activation is linked to a mass. So if you have a strong activation, you have a big mass, and this activity of the brain is curving the space-time, so directing where the action potentials flow. And where's the action potential flow, they activate the nodes. They curve the brain space-time. So it's exactly as in the universe. And you can also model with that. I mean, that's my simulation shows that you can, with this D star, this pseudo-diffusion coefficient, depending on the value, you have the short connections and the long connections. You can obtain results very similar to the connection which have been observed in vegetative patients, fully awake people, and so on. So there is a gradation of connection. I get exactly the same pattern.
Interesting.
So as a proof of concept, I said, if now with my simulation, I overactivate one area of the brain, could I suddenly reactivate a network? That's what my equations show the relativity equations. But I did the experiment in rats. So I talked before about these rats anesthetized, so it's like unconscious.
Yes.
So when you stimulate the central medium nucleus of the thalamus, suddenly the rats get awake. So basically what you do is that you resuscitate all the connections between the different brain areas. So each brain area is working independently, but by doing that, you connect regions which were not connected.
Okay.
So this is what this model is suggesting. So you see, it's far-reaching. And in the Einstein equation, he added in 1917 what is now called lambda, the cosmologic constant, because at that time, his theory was predicting the universe could extend or collapse. To have a stable universe, it did that. But later, it was discovered that the universe is expanding. So then he said it was a big mistake to have this constant. But if you work this constant in my model, this constant plays a very important role. It is explaining how different brain connections interact. So basically how social interactions take place in one brain. So you see, I mean, it's a full book I'm writing on that.
Yeah.
You cannot stop me when I have an idea.
I'm looking forward to reading that, definitely.
But what I hope, as I told you, for the IVIM, for diffusion MRI, I had a lot of critics, which were good because it helped me to make progress. But, it's like neuroscience, as we know it today, people are trying to explain the functioning of the brain, either using mathematical models, such as Flintstone, for instance, with the free energy, or looking at all the small action potentials and trying to model the activity of everything. And what I usually say to this is that my model doesn't care about what's going on in individual neurons. And the example I say is that to predict, to forecast the tidal waves in the sea. So there's a high wave, and you have the Newton's equation, Einstein's equation. You don't care about the movement of each water molecule. In fact, you cannot even do it because there are so many molecules that you don't have the power in terms of calculation, and you don't even have the models. So we don't understand what is liquid water very well. So this means that we need, of course, to understand how neurons are connected and how they work and all of that. That's very important. But at the same time, to have a global approach, it's like a dual concept. It's not in opposition.
I know. It's complementary in the sense that you can't completely from bottom up. You have to sort of capture.
But where I am afraid about how our colleagues will react to that, there is a saying that, with general relativity is the physics of the universe and so on, and quantum mechanics is the physics of nuclei, atoms, and so on. But in black holes, for instance, I talk a lot about that in my book. At some point, you have to mix the two. You have to play with both. But nobody has succeeded so far in a convincing model in physics. But Brian Greene said, when you want to talk about general relativity in a workshop dedicated to quantum mechanics, it's the same as lighting a fire camp in an explosives factory.
Okay.
So I hope this will not happen with my model.
Well, it's going to be interesting to see how it evolves. Right. Definitely.
It's just a suggestion, of course. I don't want to say it's correct everything, but it's just a new way to look at as a brain connectome, at least.
We'll leave that at that. And one thing I just wanted to circle back to before we conclude, and that is just sort of the thought of, so you're going to high field, and you're constantly pushing the envelope of what MRI could do. So what is your vision? What do you think is, what will MRI look like in 20 years or 40 years or fMRI for that matter? What do you think are the limits?
Well, I don't know who said the problem with the forecasting is the future. Of course, it's difficult, but the immediate, I would say, goals, I think, is to scan the brain at a mesoscale, where we can identify. My idea is that we have an alphabet of basic function on the cortex, right? So there are columns, there are layers, but this organization is well understood in the visual cortex, but not so much in the other areas of the brain. So if we could have some ideas about why that areas are different functionally, linked to something which is different about the cells are organized in 3D, like in the DNA molecule, and it's a special organization of the DNA which is encoding something, right? The genetic code. So I believe that we have, I don't know how many, 100, 200 basic functions which are genetically programmed, and the cells are organized in such a way that they do this job. For instance, QR codes. It's trendy now, even for COVID in France, to check your vaccination status and everything. So everybody knows now what is a QR code. But you cannot read a QR code. A human brain, the humans have designed the QR code, but they cannot read it.
Yeah, you can't.
It's only computers, because we don't have the circuits. And even if you learn, you cannot do it.
Yeah.
So,
well, that's interesting.
So you see, there are only some things we can do. Now, if you give an iPhone or a Galaxy or whatever brand to a kid, very quickly, it can make sense of the smartphone and make something about it. But there is no special area for smartphones yet in the brain, and certainly no genes. So it's just the combination of these different elementary blocks which are used together through the fibers. So we need also to see the fibers, the connections at a very high level. Increasing spatial and temporal resolution, I think, is a priority. And the dream is if we could see activation flowing in the fibers. And I think your work on the Lorentz force might be a way to do that. But I think to succeed, we need also to have very high spatial resolution. We usually consider space, but time is also important. And something else is spectroscopic imaging. So looking at sodium, oxygen 17, you have to inject, you looking at metabolites, neurotransmitters. So far, the images are, when you do imaging with that, it's really coarse.
Right.
But we might get images with the resolution we have today with water at 1.5 Tesla by going to 11.7. So I think spectroscopy might get a chance to be more useful than it is today.
Right. You need sensitivity, and I think there's a lot of room to grow in sensitivity with spectroscopy. But like resolution, even it seems that you're back to diffusion again. You're up against this limit, like maybe of 50 microns in some sense, because of the diffusion limit.
Yes. Yeah. Anyway, that could be useful. Yeah. And then there is hopefully a big surprise. I think it's Freeman Tyson who said that the revolution, the tool-driven revolution, is to find new things which will have to be explained. Yeah. I like this.
Yeah.
So my great hope is that we will discover something that we have no idea, like for diffusion, acute stroke, and what diffusion anisotropy in the white matter. That was completely unexpected. Yep. So I really hope that with your and magnet and our magnet, they are brother magnets or sister magnets. We will find something that we have no idea today, and then that will occupy many people to understand what we see.
And that's an important point with all of, it seems like MRI has sort of been driven by that, like technology, just sort of continuously advancing and trying new methods and applying new things and then finding new phenomena. And then, so it's very technology driven in that regard. Okay. All right. Well, very last question. There was so much more to get to, but you have so many other interests. But I think, like you just to mention briefly, you're accomplished piano player, you're accomplished photographer, and you've even had a hobby in developing weather forecasting, and so which was all really impressive as far as that's concerned.
Yeah, many hobbies.
Yeah. So just to conclude then, what would be your advice to young researchers starting out? They're trying to get a sense of, what sort of tactics.
I think, that's good. I'm not a philosopher, okay? So my first advice would be, you should stay in your mind as a kid. Ask questions that nobody is asking, like, why is the sky blue?
Yeah.
Well, we know, and this is how I started the weather forecasting, by the way, because in the book, my grandfather was a gardener, and in this book it was written, when the sky is red at the end of the day, it should be nice weather the next day.
Yes.
I was shocked. How could that be true? It is true. But I had to go to buy and read the many books and high levels. This is how I learned the mathematics and differential equation by myself, to understand that. But it is true. It is true. So be a child and keep your eyes open. That's the priority. Then don't take for granted everything you read, especially now with internet. You can find anything you want, and dogma. So if you feel that there's something which is weird, or try to understand and develop. But maybe another advice, which is basically a lesson that I learned by myself, is that you should be persistent. If you believe you are right and you have a great idea, don't give up. Even if people are caught attacking you or saying you are wrong, and if maybe you are wrong, okay, if you know you are wrong, you should stop. immediately.
But if you're not convinced yourself, you should just keep being.
But if you think, no, I'm sure I'm right, and then I could see even for diffusion, people say, no way. I remember, I will not say the name, but some people, when I gave talks at very beginning in the 80s, and top level scientists, no way, no, you cannot measure diffusion in the brain, blah, blah, blah. And a few years later, they were all working on diffusion MRI. So IVIM to 20 years, diffusion fMRI is still not accepted by everybody. My new theory will take half a century probably. So, so if you believe you have ideas which are correct, don't give up and be persistent. That would be my 3 advices.
That's great advice. Keeping childlike and don't take the dogma, because sometimes we just agree on things and we don't collectively, and we don't know why. And then you come in and you realize, oh, that wasn't really the case anyway. So and then persistence. I love it. I love the just amazing advice. And okay, all right. Well, this was awesome, and I really, really thank you for spending the time to talk about all your work, to give the field a little bit of sense of history and the early days with diffusion, the B factor, and everything up to IVIM, diffusion changes with activation in the future. So thank you very much for coming on. I really appreciate it.
I thank you a lot, Peter, for this kind invitation. I enjoyed it a lot. I mean, you know how to ask really great questions and to push people in the limits. So thank you so much. And it was really enjoyable.
Oh, well, thank you. Thank you.
Neurosalience is brought to you by the Organization for Human Brain Mapping. This week's episode was produced by Ekaterina Dobrikova and Kevin Setek.